Hello, today we're going to actually spend a little bit of time focusing on how DNA is used in forensics and crime solving. The topic of DNA in forensics and crime solving is fascinating and is an evolving area. The earliest documented use of DNA in identifying an unknown suspect was in 1986, where the geneticist Dr. Alec J. Jeffries had identified that DNA has patterns in certain regions that are unique to each person. He was asked to profile the DNA of a suspect in the rape and murder of a 15-year-old young woman. After profiling the suspect DNA, police collected samples from as many potential suspects as they could, and they finally found a match. In the 1990s, the use of short tandem repeats, we'll call them STRs for short, became the gold standard for DNA analysis. These are areas of repetitive DNA sequences with varying numbers of repeats and are referred to as STR loci. Regions with short tandem repeats are highly polymorphic. In this case, the alleles are differentiated by the number of repeats in the locus and mainly occur within non-coding regions of the genome. The figure is showing the varying numbers of repeats in the same locus, but in different people. See how person 1 has 5 repeats, person 2 has 6 repeats, and person 3 has 7 repeats. The process of identifying individuals based on unique patterns in their DNA is called DNA fingerprinting, and most commonly uses short tandem repeats to do this. The use of STR fingerprinting in forensic investigation has been well supported with funding, infrastructure, training, and building up of databases such as CODIS, where unknown DNA profiles can be compared to known DNA profiles. There are millions of STR regions in the human genome, but the CODIS database was built beginning with a core of 13 short tandem repeats, or STRs. When all 13 CODIS core loci are tested, the average random match probability is rarer than 1 in a trillion among unrelated individuals. In January of 2017, the number of loci for new CODIS profiles was increased to 21, including the sex marker amelogenin. This figure shows the original 13 listed in yellow, and then the 7 that were added in 2017 are in green. So here we have a case that illustrates how traditional STR-based DNA profiling is used to identify suspects. Following a double homicide, DNA was collected from multiple sources, including the victims, a rope, and other crime scene evidence. Analysis revealed an STR profile belonging to an unknown male, but when this profile was searched against the National DNA Database, there was no match. Note that the DNA found on the rope, the gloves, and the suspect DNA match at all the pairs. Several years later, during the investigation of an unrelated crime, a suspect was identified and his DNA profile was entered into the database. At that point, a match was found between his STR profile and the DNA recovered from this earlier double homicide. This ultimately allowed investigators to link that suspect to the original crime. So in this case, STR profiling was effective, but only after the suspect's DNA was entered into the database, which this highlights one of the key limitations of this approach. While STR profiling has been the gold standard in forensic DNA analysis, it does have some important limitations. It requires relatively good quality DNA. It depends on existing database matches, like we just saw in that case that we looked at. And it can struggle with complex or mixed samples, meaning, if you have multiple people or, just other environmental DNA that's mixed in there, it can really kind of muddy the water there. It also provides very limited information beyond identity. And in some cases, like with identical twins, it can't distinguish between the individuals at all. So the question becomes, what do we do when STR profiling doesn't give us an answer? Over the past decade, new genomic approaches have emerged that don't just match DNA. They help us identify unknown individuals. These newer approaches go beyond simple matching. Some methods, like using next generation sequencing, which provides much more information than STRs, and it allows for things like ancestry, inference, and phenotype prediction, as well as probabilistic genotyping, which uses advanced mathematical algorithms and computer simulations to interpret complex, low quality, or mixed DNA samples in forensics, improve how we analyze DNA. But others, like forensic investigative genetic genealogy, or FIG, and forensic DNA phenotyping, or FDP, actually help us generate leads when no suspect is known. Today, I'm going to focus on those two because they represent a major shift in how DNA is used in investigations. The primary goals of forensic investigative genetic genealogy, or FIG, are to resolve cases of unidentified human remains and long-standing violent crimes. There are approximately 24,000 missing persons and over 14,500 unidentified remains of people in the United States. There are also over 350,000 homicides that have gone unsolved since the 1960s in the United States, and currently only about 50% of murders are being solved. This is part of what's driving the development of newer approaches like FIG. So, how is FIG different? Instead of asking, does this DNA match someone? FIG asks, does this DNA match anyone's relatives? Forensic investigative genetic genealogy is a method that uses SNP-based DNA data to identify unknown individuals by finding genetic relatives in genealogy databases and reconstructing family trees. What that looks like: investigators collect DNA evidence from a crime scene, and then they create a SNP profile. The SNP profile is uploaded to a public genealogy database. There are four main databases that law enforcement use. They are GED Match, Family Tree DNA, DNA Solves, and DNA Justice. Users of these websites must consent to allowing law enforcement to utilize their DNA profiles. GED Match, in particular, plays an important role in FIG implementation. They offer an array of DNA comparison and analysis tools. Law enforcement used GED Match to identify the suspect in the Golden State Killer case. The database automatically compares an uploaded DNA profile to other profiles in the database and identifies matches. These matches are based on how much DNA is shared, which is measured in something called centimorgans, or cm. These shared segments represent something called identity by descent, or IBD, meaning DNA inherited from a common ancestor. The more centimorgans two individuals share, the more closely related they are. Even when there's no close match, investigators can identify distant relatives such as third or fourth cousins. From there, genealogists build out family trees, working across multiple generations to identify individuals who could plausibly match the suspect based on factors like age, location, and known history. This process narrows a large pool of people down to a small number of potential suspects, who can then be tested using traditional STR profiling for confirmation. It is important to mention that while forensic genetic genealogy is helpful in identifying suspects, STR is still used to confirm a match, and all normal investigative work must still be performed. For example, a suspect may be identified via genealogy, but then the suspect or the detectives must go back and determine if there is real evidence to link that person to the crime. This figure shared here represents just the workflow of how FIG works. I just included it so that you have a visual representation of what that looks like, but you don't need to know the figure for the quiz or the exam. To better understand how FIG works, we need to understand how relationships are inferred from DNA. When two individuals share DNA, we measure that in units called centimorgans, or cm. The more centimorgans two people share, the more closely related they are. This chart, which I got from a website called the Genealogist.com, shows the typical amount of shared DNA across different types of relationships. Genealogists can use this to identify the potential relationships that exist based on their centimorgans or the shared DNA. For example, siblings share a large amount of DNA, while more distant relatives like third or fourth cousins share less. When a DNA profile is uploaded into a genealogy database, the system returns matches along with how many centimorgans are shared. Investigators then use charts like this to interpret those matches. For example, if a match shares around 200 centimorgans, that could correspond to several possible relationships, such as a second cousin or a more distant relative. Because there's overlap between these ranges, FIG does not give a single answer. It provides a set of possibilities. Genealogists then use this information along with public records to build family trees and narrow down potential individuals. So, notice how much overlap there is between relationships. That's why this process requires both genetics and genealogy. For example, a sibling could range from 1,613 to 3,488 centimorgans. And then a niece could range from 1,201 to 2,282 centimorgans. So if you have a person with 2,000 centimorgans that are shared or that match, they could be a sibling or they could be a niece or nephew. Other information, such as the person's age, the location they live in, etc., can then be used to help try to distinguish what the true relationship is. Let's take a look at FIG in action. The first cold case solved using FIG, the FIG method, involved the Golden State Killer. I think we've all probably heard about that, and if you haven't, feel free to go look it up. And while forensic genetic genealogy is often used in cold cases and identifying unknown remains, the Brian Kohberger case represents a situation where FIG was used effectively to solve a current crime, one where the perpetrator could have possibly offended again if not caught quickly. In late 2022, 4 college students were found murdered in their shared home. Police were desperate to find who was responsible. Investigators recovered DNA from the crime scene, but there was no match in CODIS, so that would be using the traditional STR method. And so they didn't have a suspect from that route. They then generated a SNP profile and uploaded it into a genealogy database. The system identified distant relatives based on shared DNA, and that, again, measured in centimorgans. And it also reflects that identity by descent, so shared common ancestors. Genealogists use those matches to build out the family tree, eventually narrowing the pool of possible individuals and zeroing in on a family line in Pennsylvania. Ultimately, this process led investigators to Brian Kohberger as a suspect. To confirm the identification, they collected DNA from his family's trash and performed traditional STR analysis, which then matched the crime scene DNA. So this case really highlights how FIG is used to generate leads, while STR profiling is still used for final confirmation. Another way DNA is being used in forensics is forensic DNA phenotyping, which is the prediction of physical appearance from DNA. It can be used to generate leads in cases where there are no suspects or database hits, to narrow suspect lists, and to help solve unknown human remains cases. Several companies perform genetic phenotyping analysis, including Parabon Nanolabs, Bodhi Technology, DNA Labs International, and Othram. Parabon Nanolabs is a company who has developed a method of generating an image of someone based on their DNA, and they have funding and support from the U.S. Department of Defense. They began with large data sets containing genetic information and measures of phenotype for thousands of subjects. And then Parabon's bioinformatics team performed large scale statistical analysis on hundreds of thousands of individual SNPs and billions of SNP combinations to identify genetic markers that are associated with a trait. Next, in their modeling phase, Parabon scientists used machine learning algorithms to combine the selected set of SNPs into a complex mathematical equation for the genetic architecture of the trait. A new unknown individual SNP data can then be plugged into this equation to produce a prediction of the trait in that individual. They call it the snapshot forensic DNA phenotyping system, which accurately predicts genetic ancestry, such as genetic ancestry along with eye color, hair color, skin color, even freckling and face shape in individuals from any ethnic background, even individuals with mixed ancestry. The picture here shows a couple of individuals who volunteered to participate in the development of this snapshot technology. Parabon developed their model for prediction and then tested it against volunteers to see how accurate it was. You can see that they were fairly accurate in their phenotype predictions. Something to keep in mind is phenotyping provides probabilistic information or predictions. It's not a definitive identification like DNA fingerprinting. FDP, or forensic DNA phenotyping, is better used for lead generation than as actual evidence. So when you don't have a suspect, you're not really sure where to start, maybe you can get a profile here and that zeroes in on a certain ancestry or maybe even the region that they're from, hair color, eye color, all those kinds of things. All right, here's an example of where forensic DNA phenotyping was used to help resolve a cold case. In 1994, a 37-year-old woman was found murdered in her home. There were no clear suspects, and for decades, DNA collected from the crime scene did not match anyone in existing databases. So traditional STR profiling was not enough to identify a suspect. Investigators then used more advanced DNA analysis, including both genetic genealogy and DNA phenotyping. The genealogy component helped build a family tree and narrow down potential individuals. And then importantly, the phenotyping component was used to generate a digital facial rendering based on the suspect's DNA. This rendering predicted physical traits like ancestry and facial features, essentially acting as kind of like a genetic witness. Investigators were then able to compare this predicted image to actual photographs of the suspect from earlier in his life, which helps support their identification. So in this case, the rendering didn't identify the suspect on its own, but it provided a visual lead that helped guide the investigation alongside genetic genealogy before the final confirmation through traditional methods. And I find it fascinating. You can see the actual photo up against their genetic rendering, and it's pretty striking, very similar. So these technologies raise several important and ethical considerations that extend across all uses of DNA in forensics. One key issue is that DNA doesn't just identify individuals, it also connects them to their families. This means that genetic data from one person can have implications for relatives who never participated in testing. There are also challenges around informed consent. Individuals may not fully control how their genetic data is accessed or used over time, particularly as databases expand and new applications emerge. Privacy is another major concern because genetic data is uniquely identifiable and can be stored indefinitely, raising questions about long-term data security and potential misuse. There's also an ongoing debate about the scope of use, where these tools should be restricted to serious crimes or expanded more broadly. Additionally, issues of equity and representation arise because genetic databases are not evenly representative of all populations, which can impact both fairness and effectiveness. Finally, there is still a need for consistent oversight and regulation as policies can vary widely across databases, laboratories, and jurisdictions. So, while DNA technologies are incredibly powerful tools in forensic science, they also raise complex questions about privacy, consent, and the broader impact on individuals and families. So, to summarize, overall, DNA has transformed forensic science, with STR profiling serving as the gold standard for identification. However, its limitations, especially reliance on database matches, have led to the development of newer approaches like FIG and FDP. These methods shift DNA analysis from direct identification to generating investigative leads through relatives or predicted traits. While these technologies are powerful, they also raise important ethical considerations that will continue to shape how they are used in the future.